Hi. Yes, Harinarana, do you have any question? Can you please tell about final proctored exam? Yes, so I will. If you, I think my screen is visible. So I will go to the pattern, total marks, pass marks. Sure. So I will go to Swayam portal. You can see you already get notification. Okay. Mm, so I am opening the informations about the course and accessing the content. So exam format October 2023. Dear candidate, this is applicable only for the exam registered candidates. So I hope you all or some of you has registered for the exam. So you will have to appear at the allotted exam, okay, exam center, and produce your hall ticket, and you have to show photo identification and all. And after that you can find the final allotted exam center details in the hall ticket. So you will be getting hall ticket soon. The hall ticket has not yet released, so you will get a notification. Type of exam will be computer based. Okay, please check in the above list corresponding to your course name. Okay, so you have, according to your course name, and the question will be on the computer. So the way you give other CBT exams, so this is also same. The questions will be on the screen and answers you have to enter on the computer. Type of questions may include multiple choice questions, fill in the blanks, essay type answers. Okay, so you will have MCQ. You may have fill in the blanks and as well as essay type answers. And type of exam is pen and okay, so you have two options: computer based exam and pen and paper exam. I think regarding your it will depend on what type of course you are having. So the questions, type of exam, the question will be the computer. You will have to write your answer on the sheet. Physical calculators are not allowed. Okay, so kindly use the link to get an idea how the on-screen calculator. Okay, if you need any calculator, you will have one on-screen calculator. And another one, I think, yeah, type of exam will be available in the list. Let us show the list. So your course name is experimental biotechnology. We have got this one, 12-week course. Exam will be on October 29th. Okay, so you have around 10 days time now. October 29th, online computer based. Okay, your exam is online, and questions will be mostly MCQ, and then fill in the blanks, but you will have essay type also. And the assessment is that around, no, it will take some time. That the same day you won't get marks. After evaluation, I think they will send notification of your marks, like you give other online exams na, NET, CSI NET, DVT, like that. So then you will get certificate on the basis of your marks. And the total marks, the assignments will also get considered. Okay, around 30% will be from the assignments, and then 70% from the main your exam. Yeah, here you will get, I think, uh, so it's around 30/70 or 25/75, like that format. And any other question? Will the questions be from study material? Yeah, definitely will the questions, all questions you have seen now, assignment questions are from all from study materials. So please go through those study materials well. And if you have, like, next week also we will be having one doubt session on 23rd, 23rd or 24th October. So that day also we'll discuss. If you just read through all those your previous classes and then tutorials, and if you have any, I scored about 80 to 90 marks in 11 assignments. Wow, that's great. That's great. So yeah, do well in the final exam also. So all the best. And if you have any specific doubt, we'll discuss. Another assignment is left, assignment 12, tomorrow. I hope you have already submitted. If not, you will submit. But for tomorrow's assignments answer, I will be discussing on next day. And next day I will be also going through doubts from all courses, like classes, all 12 classes. So today I don't have any study material to study, because we have already covered assignments, assignments as well as study material from week 4. But today I'll be just discussing last one assignments you had due, and if you have any specific doubt, I'll be discussing. So I will directly go through as week 11 assignment or quiz. So the question was, what is the purpose of dideoxynucleotides? I hope you can visualize my screen. What is the purpose of dideoxynucleotides in Sanger sequencing? Dideoxynucleotides, that means not only one deoxy but also dideoxy, that means 2 OH group is 2 oxygen is missing. So question is, what is the purpose of dideoxynucleotides in Sanger sequencing? Options: to elongate the DNA strand during synthesis, to initiate DNA synthesis, to terminate DNA synthesis at specific positions, to label the DNA fragments for detection. So dideoxy is definitely not to elongate the DNA strand, because if you don't have OH group at 3 prime, then that won't be able to get synthesized or get elongated. Also not to initiate DNA synthesis, this is also not true. To terminate DNA synthesis at specific position, so this is the correct answer, that to terminate. And okay, so and the next question is, which component of the Sanger sequencing reaction allows for the visualization of the separated DNA fragments? Which component of the Sanger sequencing reaction allows for the visualization of the separated DNA fragments? So the options are fluorescent primers, radioactive labels, ethidium bromide, fluorescently labeled double deoxynucleotides. So which component of the Sanger sequencing reactions allows for the visualization of the separated DNA fragments? It will be fluorescently labeled dideoxynucleotide triphosphates. What is a typical error? What is a typical order of So, what is the typical order of steps in the Maxam-Gilbert sequencing method? What is the typical order of steps of Maxam-Gilbert sequencing method? So, options are chemical modification, gel electrophoresis, DNA cleavage, visualization. DNA cleavage, chemical modification, gel electrophoresis, visualization. Gel electrophoresis, chemical modification, DNA cleavage, visualization. Visualization, chemical modification, DNA cleavage, gel electrophoresis. Okay. Maxam-Gilbert sequencing method. So, if you go through Maxam-Gilbert sequencing method, the method was discovered by Alan Maxam, and it's based on what happens here. 3' or 5' radio level DNA is treated with the base specific chemicals which randomly cleaves DNA at their specific target nucleotide. Okay. These fragments are analyzed on a high resolution polyacrylamide gel and autoradiogram is developed. So, at first, base specific reaction. Different base specific reagents are used to modify the target nucleotide. At first, reaction 1, dimethyl sulfate modifies N7 of guanine and then opens a ring between C8 and N9. Reaction 2, formic acid acts on purine nucleotide G plus A reaction by attacking on glycosidic bond. Reaction 3, hydrogen breaks the ring of pyridine T plus C reaction. Reaction 4, where as in the presence of salt, it breaks the ring of cytosine. Cleavage reaction, after the base specific reactions, piperidine is added which will replace the modified base and catalyzes the cleavage of phosphodiester bond next to the modified nucleotide. Interpretation of the band in autoradiogram. G reaction, G plus A reaction, T plus C reaction and C reaction. So, the typical order will be, at first you have to cleave your DNA cleavage, then chemical modification. After chemical modification, you have to run gel, so gel electrophoresis and then visualization. Next question, what is the primary purpose of using DTT in protein sequencing? Options, to determine the protein secondary structure, to cleave peptide bonds between amino acids, to reduce disulfide bonds in the protein, to determine the protein's molecular weight. So, DTT used as a reducing agent which reduces disulfide bonds if those are present in your protein. So, the answer is option C. Question number 5. Cyanogen bromide cleaves the protein sequence after which amino acids? So, we have read in previous class. So, we have covered this in previous class that after methionine, cyanogen bromide cleaves the protein sequence after methionine. Which of the following is Sanger's reagent? Options: 1-fluoro-2,4-dinitrobenzene, 3-fluoro-2,4-dinitrobenzene, 2,5-difluoro-2,4-dinitrobenzene, 2-fluoro-2,4-dinitrobenzene. So, the Sanger reagent's chemical name is 2-fluoro-2,4-dinitrobenzene. Okay, 2-fluoro-2,4-dinitrobenzene. So, Now the next question, what is the result of the Edman degradation method in protein sequencing? So why do you do the step of Edman degradation during protein sequencing? Options are: it breaks peptide bonds to release individual amino acids, it determines the secondary structure of the protein, it cleaves disulfide bonds in the protein, it analyzes protein expression levels. So option is, it breaks peptide bonds to release individual amino acids. Okay. Uh, what role does a carboxypeptidase enzyme play in protein sequencing? It cleaves the N terminus of the protein? No. It cleaves the C terminus of the protein. It identifies post-translational modification. It cleaves modified amino acids. So carboxypeptidase means it cleaves the protein at its C terminal. So what is the following is a common method for detecting RNA hybridization in northern blotting. So during RNA hybridization in northern blotting, we have covered in previous classes that after transferring, what we do autoradiography to detect whether your RNA of interest is there or not. Okay. Next question, what affinity column is used for purification of mRNA? So as we know that mRNA has poly A tail, so we use poly T affinity column so that those adenines will now bind to thymine and then we can elute it further. So option is poly T. What is the significance of pre-wetting the nitrocellulose membrane before mRNA transfer in northern blotting? Options: it prevents RNA degradation, it reduces background noise in hybridization, it increases mRNA electrophoretic mobility, it enhances mRNA binding to the protein. So basically it enhances. So whenever we are doing pre-wetting the nitrocellulose membrane, your transfer will be efficient as it enhances mRNA binding to the membrane. In northern blotting, at what temperature do you develop your autoradiogram? So it's minus 80 degree. That temperature you will develop your autoradiogram. After RNA transfer in northern blotting, what is typically done to fix the RNA on the membrane before probe hybridization? So you have to, after transferring your RNA, you have to do UV cross-linking and then you will be proceeding towards probe hybridization. What is the primary purpose of southern blotting in molecular biology? Options: to analyze protein-protein interactions, to determine the sequence of a DNA fragment, to detect and analyze specific DNA sequences in a sample, to quantify RNA expression levels. Southern blotting, the answer will be to detect and analyze specific DNA sequences in a sample, specific DNA fragments in a sample. So I think there is a controversy, there is some mismatch in the options. To determine the sequence of a DNA fragment, no, it should not be the answer. The answer is giving this. So answer will be to detect and analyze specific DNA fragments in a sample. Which of the following is an advantage of vacuum transfer over capillary transfer in southern blotting? Which of the following is an advantage? Okay, so what is the advantage of vacuum transfer over capillary transfer? Options: faster transfer times, obviously. Second, lower cost of equipment, no. Better control over transfer conditions, no. Less risk of sample contamination, no. So the main advantage is that it's very much faster than capillary transfer. Okay. Do you have any specific questions? Then you can write down in the comment section. I will go through. So we have covered assignment 11. Assignment 12 is live and your deadline is tomorrow. So I won't be able to solve questions for you. but those questions, if you read through, you will be able to understand that those all are from your study materials only. So week 12 assignments are mainly from, I think, last 2 to 3 classes. So go through those and then try to solve those answers. Next class we'll be discussing possible like answers of these assignments. And for now, if you have any doubt, you can write down in the chat section. We'll be happy to discuss.